Before I invite our first group of students to talk, just a few special mentions to some of the people who made this summer school possible. So we've had a couple of facilitators who've been really great, Jamie Gallagher and Charlie Ashby-Roberts. Our careers panel, who joined me this afternoon, Dr. Martin Clift, Dr. Danny Amovia, Dr. Ovi Novak and Laura Bramwell. Thank you so much for joining us. That's really fascinating. And huge thanks to Rebecca Shepherd and Darcy Wilson for organising the summer school, to Mimo for organising this event, and to Jared for overseeing everything. Okay, so without further ado, I'm going to hand back to Jared to introduce our first group of students.
Thank you very much, Colette. The first talk is by Riddhi Sharma, who has developed a method to study the genetic and environmental causes of Parkinson's disease in human brain cell lines to assess whether the cells are entering a diseased state at Public Health England. Over to you, Riddhi.
Thank you very much. Lovely. Can everyone see that? Wonderful.
Yes. Yes.
All right. Perfect. Okay. So hi all. I'm Riddhi and I'm an undergraduate student studying pharmacology at the University of Bath. I completed my summer studentship with Public Health England, as Jared mentioned, and today I'll be taking you through our project on the production and potential use of an epigenetic tool in Parkinson's disease. So in the long run, we are actually hoping that this tool might actually be used not only in neurological conditions, but stretching out into other fields such as oncology. So the central idea of our project revolved around using CRISPR, a technology used to target and edit genes, and deactivated Cas9, an enzymatically inactive form of CRISPR-associated protein 9, alongside an effector protein to control the expression of a particular gene. So the effector protein that we used in this case was P300, which is a histone acetyltransferase, allowing the increase of the expression of a particular gene. So we decided to target the gene responsible for the production of TPPP, tubulin polymerization promoting protein, try saying that 5 times fast, is normally found in oligodendrocytes in a healthy person, but can become localized in neurons as well as oligodendrocytes in the Parkinson's disease state. So our main tool was a lentivirus, which I promise isn't as awful as it sounds. So you may recognize the term lentivirus from the well-known disease HIV. But we actually decided to use lentiviruses for the good, as they are able to hold larger plasmids than other viruses, such as adenovirus, and due to their ability to modify and be incorporated into the genome. So we altered various plasmids to contain the guide RNA for the TPP coding region, and to contain the coding sequence for the P300 effector protein. So due to issues with the size of the plasmid, we ended up actually modifying our original plans to produce a singular plasmid with all of the necessary components in it. Along the way, we made substitutions for animal-derived products, such as supplementing cells with GrowPro, as opposed to the commonly used fetal bovine serum, and by replacing trypsin with a physical cell scraper. By replacing crucial components such as these, I was able to understand just how often animal-derived products were actually used in research and how important it is to move towards synthetic replacements for them. So unfortunately, due to technical issues that arose, mainly around contamination of cell cultures, I did not actually have enough time to complete a successful transduction of the third plasmid into the cells, but I actually plan to continue with this and plan on doing this over the next few weeks. And I will hopefully be able to analyze the levels of TPPP and of the RNA. So in order to measure protein levels, we'll be using immunoprecipitation. And in order to check RNA levels, we'll be using qPCR. I'm very much looking forward to the next stages of this project, and I'm super grateful to receive the opportunity to learn more about not only neurobiological diseases like Parkinson's disease, but also for the new knowledge that I've actually gained in the animal-free research side. So thank you so much for listening, and any questions would be welcomed.
Thank you very much, Riddy. Just so everyone knows, we're going to have questions after the first set, after each of the sets of 5 presentations. The next presenter is Izzy Breithaut, who created, characterized, and adopted an animal-free approach towards genotoxicity testing of a variety of different human toxicants using advanced human lung and liver model systems at Swansea University. Over to you, Izzy.
Hi, I'll just start sharing my screen. Okay, is that on everyone else's screens now?
Looks good, Izzy.
Yeah, okay, great. So I am studying pharmacology at the University of Liverpool, and I did my placement at Swansea University. The aim of my experiment was to determine whether synthetic serums can be used instead of animal serums to grow cells to then be used for genotoxicology studies. So I grew TK6 cells, which are essentially a form of lymphocyte, and A549 cells, which are cancerous lung cells. I cultured them both in synthetic serum, and I compared the data that I had with historical data of cells that were grown in animal serum. So I didn't use any animal products in my own research. So after growing both of those cell types, I exposed them to 3 doses of potassium bromate and 3 controls, and then recorded the oxidative DNA damage that is caused by potassium bromate using a micronucleus assay, which is where you count the number of micronuclei, and the more micronuclei that I count, then the more DNA damage has occurred. So I was able to determine whether the synthetic serum can grow cells that can be, where the results could be comparable to when they're grown in animal serum. So the results that I was getting in were more complex than a simple yes or no about synthetic serum being able to be used. I was becoming more and more intrigued in the results as I was getting the data in. So I found it really rewarding doing this entire project. So I found that with TK6 cells, synthetic serum makes them less sensitive to the effects of potassium bromate. So I recorded the cell survival and the number of micronuclei. So in TK6 cells grown in synthetic serum, I was finding that they were surviving more than we would expect, and the number of micronuclei caused by the potassium bromate was lower than expected. So this made the TK6 cells not really a fantastic cell type to be grown in synthetic serum for genotoxicology studies. However, the A549 cells were had much more promising results, and they were much more as expected. So it meant that the A549 cells were suitable, which is fantastic because it potentially means that we can stop using fetal bovine serum to grow A549 cells for toxicology studies and use synthetic serum instead. So my aim was fulfilled, which was amazing, and I felt really proud that I had collected the data for that. And if I had to conclude my whole presentation into one sentence, then it would be that synthetic serum can be a successful alternative to animal serum, but it is dependent on cell type. But even the dependent on cell type part is still really positive because it's still a massive step forward in the right direction to an animal-free future. So yeah, I'd be more than happy to answer any questions. I had the best time doing this project, and I found it a lot more interesting than I originally thought. I didn't really know what to expect when I first started my project, and it was amazingly rewarding, and I gained so much experience. That's everything.
That's fabulous to hear. Thank you so much, Izzy. Can I just ask, before you mute yourself, before we move on, I don't know if I missed it, but in your practice yesterday, you said why you were testing potassium bromate and where you find it. And I'm not sure I heard it this time, and it came across as one of the really interesting points of your talk. Would you like to tell everyone?
Potassium bromate is actually used in the production of bread, so it's such a simple, common compound. Yeah, so yeah, it was really interesting because you just wouldn't think of it really.
Yeah, I didn't know that, so I thought that was a really interesting point. So thank you so much. Next up we have Morwenna Oldfield, who built a cell-based membrane, which will act as a model for the cornea, allowing her to find novel ways of delivering drugs without relying on testing in animals at the University of Birmingham. Welcome, Morwenna.
Hi, let me just share my screen. Can everyone see that?
Yes.
Okay, perfect. Hi everyone, my name's Morwenna and I did my summer project at the University of Birmingham. So common therapies for eye diseases such as age-related macular degeneration include techniques like monthly injections of anti-VEGF antibodies into the eye. And although this is an effective therapy in combating this really common disease, it does involve various adverse side effects like uveitis, which is basically inflammation of the eye. The lab group I was working with over the summer, they've designed an effective novel eye delivery system that delivers drugs into the eye without any of the additional side effects. The delivery systems use peptides as delivery agents to deliver the drugs throughout the eye. However, methods to determine the efficacy of these drug delivery agents have involved using animal products or resources. So things like pig eyes or even simple things like chicken eggs. So during my summer project, I managed to test the efficacy of these drug delivery agents in delivering Avastin, which is a common drug used to treat eye diseases, whilst only using animal-free resources. So to summarize my work over the summer, I've split the work into 4 main stages. The pictures with the corresponding numbers are the results I generated, like a summary of the results I generated from each stage. So firstly, I grew primary human eye cells to confluence in animal-free media. Growing the cells to confluency meant I could create a human cell monolayer that acted as a membrane which simulated the cornea, which is a tissue in the eye. And the cornea is one of the hardest tissues for the drugs to actually be able to penetrate through. This process was the process that I personally found the most challenging because I'd never done cell culturing before ever, and everything kept dying, everything kept going wrong, and I was so ready to give up with it. I hated it, and then eventually everything started growing to confluency, which was great. And so this then led me to completing the second stage of my project. By adding Avastin to a fluorescent dye, I was able to see the natural level of permeation of Avastin throughout the membrane without any drug delivery carriers in place. And as you can see from picture 2, results showed that the cell monolayer membrane was impermeable to just Avastin and the fluorescent dye on its own. So next, in the third stage of my project, I determined the efficacy of the actual delivery agents themselves across the cell monolayer membrane. And as I've said previously, the delivery agents are various peptides, in particular R6, CC12, and penetratin. And results show that these peptides can cross the cell membrane on their own. And then finally, using an ELISA, I found that the cell penetrating peptides were able to effectively deliver Avastin across the membrane. So these results have shown not only that the peptides will be able to penetrate through the tissues in the eye and be able to deliver various treatments to combat different eye diseases, but also that the testing of the efficacy of these methods can be done in vitro without using any animal-based resources at all. Yeah, I had a great time over the summer. I'd really like to thank everyone for giving me this opportunity and for not only letting me develop my lab skills, but also for raising my awareness and knowledge of what we can actually do in animal-free research. Thank you for listening.
Thank you so much, Morwenna, and I'm sure it's worth saying again, and this cropped up in practice yesterday, that Morwenna was really honest, and anyone like myself and like other people listening who's ever done tissue culture can identify with this. There are times where you have infections and you just really struggle to get rid of them and to get back on track. And Morwenna was really honest about that. And what was wonderful was just what came across Morwenna was your tenacity and your determination to get over that and get things sorted and get your results, and you did that. So that was a really nice, honest and lovely part of your talk. So so well done indeed. Okay, next up, we have Amir Fard, who used 3D human organoid models of normal colon tissue and of colon cancer to help to identify new treatment strategies at Cambridge University. So thank you and welcome, Amir.
Hello. So I spent this summer fascinated by a specific type of colorectal cancer, which is known as 3Ks. And these 3K cancers encompass a group of colorectal cancers defined by specific mutations in TCF7L2, APC, and unusually for right-sided tumors, they never contain BRAF V600 mutations. So going into it, I plan to gain a better understanding of the characteristics of 3K colorectal cancers with the hopes that this would allow us to better manage them in the future. And as I read more about these types of colorectal cancers, I found that their colorectal cancers are generally classified into 4 main groups, and these are known as consensus molecular subgroups, or CMS for short. Now, it's rather elegant being able to sort thousands of colorectal cancers into just 4 subgroups, as it means that management can be more personalized. However, not every colorectal cancer can be generalized under these 4 subgroups, and I hypothesize that 3K's colorectal cancer is an example of this. To find out if there was any truth behind this, I analyzed single-cell RNA sequencing data, and this data set essentially provides us scientists with insight into which RNAs are found in a cell, and hence which genes are being expressed. And rather excitingly, preliminary data does indeed suggest that 3K colorectal cancer does not fit into any of the 4 previously described groups. The importance of this to us researchers and clinicians is that identifying a patient with a 3K colorectal cancer suggests to us that they should be managed uniquely rather than using management plans that would otherwise be prescribed for the other 4 subgroups. So the second aspect, So the second aspect of my project led me to Addenbrooke's Hospital, where I was fortunate enough to shadow a histopathologist who introduced me to H&D slides. And working through real patient cases, I learned how to identify and stage different types of colorectal cancers. I'm extremely grateful for this opportunity and found that as a hopeful clinician, this marked an elegant example of where clinical medicine meets clinical research. From a research standpoint, being able to create links between clinical parameters, for example, how mucinous a tumor is or what stage it may be, and the genotype-phenotype work that I had done earlier may allow us to better identify colorectal cancers that are less defined either in terms of their histology or their gene expression programs. So essentially, by making these links, we can better classify colorectal cancers that may have been previously more challenging to do so. And the last, and for me, most interesting aspect of this summer was learning how to grow and manipulate organoids. And these organoids are essentially really cool models for cancer in a tube. And my lab has pioneered an unprecedented novel organoid culturing technique that is free of animal-derived products. And this ties in quite nicely with my passion to find animal alternatives. So what this essentially means is that we can conduct our research sustainably without endangering any animals in the process. One type of manipulation of these organoids is known as a transfection, whereby a gene of our choosing is effectively implanted into these organoids. The gene we chose was Fuki, which is a cell cycle reporter that allowed us to define site-specific cell division in these organoids. And essentially, we are analyzing the rate of proliferation of cells within our organoids, as they'll rather elegantly cycle between red, orange, and green fluorescence, which we can measure. What's more is that we can look at which cells spatially within the organoids are proliferating most, and this can provide researchers with key insights into the validity of organoids as cancer models. So I carried out transfection using 2 protocols: electroporation, where electric pulses help the cells to take up our gene, and lipofectamine, where our gene is contained in a lipid capsule that is delivered to our cells. And this was useful because we were able to compare the efficiencies of each transfection for my colorectal cancer cell lines. And what we see in the most right image on the final panel is an organoid that has been successfully transfected, with arrowheads showing specific cells expressing our Fuki gene. And adjacent to this is an organoid which hasn't been infected, and so it can be considered as a control. Most importantly, my research has successfully shown the application of organoids without the use of animal-derived materials, which going onward suggests a more sustainable and positive future for colorectal cancer research. And before I end, I'd just like to thank everyone for this opportunity, particularly my supervisor, Mark Delaroche, and Animal Free Research. It's really been eye-opening, and yeah, it's really kind of cemented my desire to become a clinician with a research focus. So thank you.
Thank you so much, Amir, and thank you for your kind words at the end, and more great research. Thank you for your presentation. Okay, we have the last of this group of 5 now. It's Ali Mahmood, who used a hybrid of computer learning and animal-free laboratory techniques to develop a prognostic marker for head and neck cancer at Queen Mary University of London. So welcome, Ali.
Thank you. Just share my screen. Perfect. Cool, so thank you everyone. So I'm Ali and I study medicine at Barts, and this summer I was working on this project, developing an aging-associated or senescence-associated tumor predictive score using artificial intelligence approaches. So the cancer we were using for this project were head and neck cancers because they're very common. They affect something like 900,000, there are something like 900,000 new cases every year around the world and 400,000 deaths. And it's the 8th most common form of cancer in the UK. So with regards to this cancer, we were specifically looking at senescence markers. So this is when cells stop proliferating or dividing, but remain metabolically active. And this means they continue to secrete a variety of factors as part of what is known as a senescence-associated secretory phenotype, or a SASP. And this is thought in the long term to aid tumor growth, although the link is not 100% clear. And this is just outlined in this image down here. So this got us asking the question of whether we could use these senescence markers to aid the diagnosis of head and neck cancers, which brings me to the aim of the project, which was to determine the diagnostic potential of senescence-associated genes in head and neck squamous cell carcinomas using computational approaches. So how do we go about this? So the first thing we did was a literature review and a database search to generate a list of senescence markers that we could investigate. And this came to a list of 411 genes. We then had curated an RNA-seq data set of 460 tumors with a matching number of controls. And we used a Python script to generate heat maps for our 411 senescence genes using that data set. And they look something like these 2 examples here. So in all of these heat maps, blue represented a downregulation of the gene and red represented an upregulation. So in these 2 examples, for instance, CCR16 here, in the cancer, it was downregulated but compared with our controls. But for MAGE-A2, the opposite is true, where it's upregulated. So we had 411 of these heat maps, so we needed to kind of filter out the significant ones. So we used a couple of approaches. So in some cases, there's a visual difference between the cancer and the controls. But we also used the software to differentiate. So we looked for significant differences in the means and the medians between the cancers and the controls. And that got us to this list of 9 genes here. So again, blue is a downregulation in the cancer and red is an upregulation. We then put these genes into some machine learning algorithms to assess how reliably they could diagnose the cancer. And using one classifier, the KNN or K nearest neighbor classifier, we were able to achieve a greater than 80% diagnostic accuracy in 7 of these 9 genes. So all of these with the exception of MAGE-A2 and RBP2. So this is really interesting because there isn't currently a gene signature that is used to diagnose this form of cancer. So it's something quite novel. So in terms of future work, we'd also like to look at prognostic markers to really establish the effects of senescence on head and neck cancers, and we'd like to validate these findings in the lab. We were planning to run qPCRs for these 9 genes, but unfortunately we ran out of time with the project. So just to conclude, we used machine learning techniques to create a signature of 9 genes which could potentially be used diagnostically for head and neck squamous cell carcinomas. I'd just like to, before I finish, I'd just like to thank everyone who was involved in this project and everyone at Animal Free Research UK. The project was like really enjoyable to be a part of, and I definitely learned a lot. So thank you everyone for listening.
Thank you very much indeed, Ali, and thank you for your kind words too. That's the first 5, so thank you for your presentations, and it's now time for some questions. I know that Mimo you were collating some questions, and you asked a very general question for everyone to answer. But I wonder if before that we got some specific questions from Jeff and from Valerie. And I wonder if we could address those first, if that's okay, before we move on to your question. So Jeff, you had a question. Yeah, you had a question for Rudy.
I did. Yeah, can you hear me?
Yes.
Yes. I can't see any of the other people apart from you. I'm not quite sure whether that's the way it should be on this presentation. So to Rudy, thanks for a very nice presentation. And it's always good to see people working in the neurosciences. And I know Amanda Ellison is also in the audience, so she may have something to say about this as well. But first of all, it's something of a question, and that's the use of the two cell lines, one of which is the SH5Y, which is a neuroblastoma cell, which is being used, I guess, in lieu of the absence of a proper neuronal cell, because neuroblastomas are neoplastic, they're tumor cells. And the other thing, HEK, out of sheer ignorance, and you can do 50 years of medical research and still be ignorant, as I am. So, feel encouraged to all you young folks out there. I'm not sure what the HEK represents, so that's a question. The second one is could you perhaps use something more representative of a neuron, a non-neoplastic, non-tumor neuron, rather than using the neuroblastoma? And I appreciate the complexity of culturing oligodendrocytes. These are the cells that produce the myelin in the peripheral nervous system. For those non-scientists in the audience, very important for neuronal transduction of messages along fibers. And then finally, you talked about the replacement of fetal bovine serum. What were you replacing that with? Were you replacing it with human serum or chemically defined serum-free medium? So there's sort of a couple of questions and a couple of comments in that. So really, can you reply to some of those?
Yeah, sure. Thank you for your questions, first of all. So to address your sorry, I can hear it. So yeah, to address your first point, which I believe was about why we used X cells. So was that your second question? I can't remember.
So it doesn't matter the hex cells. What are they? And the second one was the neuroblastoma cells. Is there anything you could use in place of those, which was actually a non-tumor, a non-neoplastic? Because these are not brain cells, they're neuronal, but they're not brain cells, they're tumor cells. They're used in many experiments and have been for a long time, but they're not really a proper neuron. Is there any other way you could have addressed that problem?
Yeah, sure. So hex cells we used initially when we were doing our virus production. So once we had produced the virus using the SVG P8.91 and using P300 and the guide RNA, we implemented that virus. We tried to transfect it into hex cells because they're quite renowned for their protein production. So obviously we wanted large amounts of virus because the plasmid that we were using was quite big. So we were already kind of pushing the limits on what the lentivirus could hold. So what we wanted to do was have as much of that kind of being pushed out into the medium as possible, so that we had a more successful rate of transfection and then eventually of transduction.
Okay, that's right.
And then in terms of the neuroblastoma, we decided to go down that line because the TPP, when it gets to late stage Parkinson's, and sometimes in early stage as well, it's actually concentrated in both oligodendrocytes and neurons. So we wanted to have sort of something that was almost like a kind of blanket sort of cell line for it. So we'd be able to observe and work with sort of slightly easier cells first and see the effect of this tool that we're creating in that. And then hopefully when we take it forward, we'll actually try and work with more specific cell types. And of course, down to timing issues and stuff, we don't want to, because they're a little bit harder to culture. So we wanted to be a bit more on the safe side and work with slightly more quickly proliferating cell types.
Hang on. Thank you very much, Rudy. I wonder if we can now, just for the sake of time, first of all, can everyone see the chat? Because Mimo's very kindly put the questions in the chat, and that'll save me having to ask them out loud. Well, not save me, save the time. So if you can look at the chat, I think what I'd like to point you to first, we could maybe go around the 5 presenters so far, and you can all weigh in on 2 particular questions. The first one is, what initially led you to seek animal-free alternatives? Was it a desire not to use animals ethically, or was it a belief that you would see better results? And what animal or animal-derived product would your project replace? So let's do that. Well, Riddy's just answered, so let's ask Izzy first, if you could weigh in on those 2 questions, please.
Okay. So the animals that my project did replace was horse serum for TK6 cells and fetal bovine serum for A549 cells. I just used the same synthetic serum to grow both of them, which was a human replacement serum. And the first question was the seeking animal-free alternatives. I'll be completely honest, when I started the project, I just wasn't that aware, as I am now, of the extent of animal use in research. I really wasn't. So entering an animal-free research project, I just thought would be the easiest way to gain proper knowledge that was reliable, really. The best way to get myself started as an ethical scientist, which is ultimately my goal. I would like to continue my career as an ethical scientist. So I just didn't know. I didn't know whether I would see better results with synthetic serum or with animal serum. So the whole project was really eye-opening, and I learned a lot more than I even thought there was to learn about animal-free approaches. I just didn't know there was so much scope for new paths that I could go down. So I'm just really pleased that I've done the whole thing, and I think I would do it all again. I really would.
Thank you so much, Izzy. As Laura Jane said on the chat, and I was going to say the same thing. That's something we hear an awful lot. So so thank you for your honesty and for that information. We'll move now on to Morwenna. Would you mind taking your turn and answering those questions, please?
Yeah. So originally, I think the way the lab group that I was working with over the summer, they were testing out these new delivery methods was by using animal eyes. I think particular was pig eyes, and they were also trying to, they were themselves trying to go away from that, and they started using chicken eggs, and so the membrane in the chicken eggs as trying to use that as the membrane stimulating the cornea. Obviously that still is an animal product. And I think just they were like, right, can we do this animal free? Also, there was the issue, not only for ethical reasons, but also technically, animal eyes, there's a lot of anatomical differences to human eyes. And yeah, so they were like, okay, cool, can we do this using a cell monolayer assay? And I was like, Izzy, I didn't really understand. I wasn't very aware of how much animals have been used in research, and to the extent of it. I remember speaking to my supervisors and them telling me how many pig eyes they used to go through and stuff. And yeah, by doing this project, it does show that you can achieve the same results, if not more acceptable, I guess, in a way, by doing everything animal free, and it's the same. And yeah.
Thank you, Morwenna. Amir, your turn, please.
For me, in terms of how I got into it, I integrated in biochemistry this year, so I'm a medic, and one of the modules we did was cancer, and within that we had 2 lectures on experimental model systems. So there the main model system essentially for cancer is GEMS, genetically engineered mice models. And I found it quite awful, just seeing it, the number of mice which are killed in order just to study cancer. And so I emailed my supervisor. So the the first lecture was on GEMS, the second lecture was on alternatives to mice models, and I emailed that lecturer and I asked, I thought your lecture content was really cool. Can I do some research with you? And he was looking at the alternatives, and so that's how I essentially got into it. And now to answer how can the research I did effectively replace animal research. Well, in two ways. Firstly, organoids. You're obviously not using an entire mouse, so that immediately gets rid of killing an entire mouse. But secondly, organoids still use some mice material. For example, the matrigel. So what you dissolve the organoids in is the ECM that's derived from mice typically. But with my lab, what I found was really interesting is it's completely animal-free derived. And so the main goal of the research for us was just to kind of show that we can grow these organoids in our new, in this novel matrigel, and grow them as effectively as has been done in matrigel, which has been derived from mice ECM, and still be able to manipulate them with transfections, etc. And we have shown that. So yeah, that's how we effectively can replace animal research by replacing firstly mouse models, and secondly replacing the use of mice ECM in the matrigel for the organoids. Thank you.
Fantastic. Thank you, Ami. I really appreciate that. And Ali, your turn, please.
Thanks. Yeah, so how I got onto this, so I was looking for a project to do, so I wasn't too aware of the extent of animal use those who answered this before me. But when I got told about animal-free research, I think that really and then doing the project as well, it really showed me how much it is used, and then it doesn't make sense kind of to use animals, especially when there are other ways of conducting the same research. And then in terms of the animals that we did that were not used, that could have been used in the project. So normally cancers are kind of grown in mice and then harvested, which is obviously it induces a lot of suffering on the mice. So obviously by using computational approaches like we did, we avoided that, which yeah, I thought was really good.
Thank you very much, Ali. And back to Riddhi now for your turn, please.
Yeah, so I've actually always, since birth, I've been vegetarian. And before it was due to cultural reasons, and then eventually it ended up being because it was just my choice and I wanted to be. So I think working in an animal-free setting was quite important to me anyway, especially because the university that I go to, they give us a wonderful opportunity, which basically means that we have our personal license, which is working with animals and doing animal research, which I can appreciate is a brilliant thing for many people to be able to have and to use. But for me personally, it was something that I did because I wanted to be able to have that qualification. But as I was doing it, I realized that actually I know that I've heard about alternatives for a lot of the things that we're doing with these, that we're going to be using these animals for. And I know that there's other stuff out there that can be used. So I couldn't quite understand and comprehend why we weren't kind of using that. And then when this opportunity with Nora came up to be working in an animal-free setting, I just kind of jumped on it almost, and I was like, that's brilliant, that's perfect. That's exactly the type of thing that I do want to go into, and to work towards science being more sustainable, environmentally friendly, and being less cruel. So yeah.
Thank you, Riddhi. We have another couple of minutes, so hopefully we can squeeze at least one more question in. There's a question from Valerie Spears, who said, why do some cell lines thrive in synthetic sera but others don't? Do we know? And is human serum an option? Anyone can weigh in on this, just wave your hand or flag yourself up. But perhaps that was a question for you, Izzy, having done that sort of thing in your project. Is that something you can weigh in?
A little bit. So the TK6 cells, they obviously didn't really, it turns out that they're not really suitable for synthetic serum growth. My research didn't go quite as far as to evaluate why, but it's definitely an area that should be looked into, because it might be that there's just a separate variable that could be altered in order to achieve the synthetic serum positive results. it could just be that my research didn't incorporate, you know, that one variable that could be changed. So obviously further research into that would be incredible, because there is potential for synthetic serum to be able to be used with TK6 cells. As for the part about humans, is human serum an option? I don't know massive amounts about human serum, but I'm kind of assuming that if I know that the way that fetal bovine serum is collected is from a fetus, so I'm presuming that there'll be quite a lot of ethical issues surrounding using human serum as well as fetal bovine serum. But obviously my research just used a human replacement serum, so it's a completely synthetic serum, and I did get positive results for the A549 cells. Yeah, that's about all that I can weigh in on that one, really.
Thanks, Izzy. It is a really interesting topic, which it would be nice to delve into further, but it is, well, yeah, Carl has asked. So seeing as Carl has asked, let's have another couple of minutes. Can anyone tell us more about human serum? My explanation was going to be just an assumption of why we use FCS or FBS instead of human serum, but somebody on here might know exactly why we don't. So if anyone would like to weigh in, please do.
So I recently presented a little bit on this at your last conference, so maybe I can weigh in a little bit. Jeff's just put an answer there saying human serum is not fetally derived and is available from accredited suppliers, but it is very expensive. So that's one barrier as to why other people wouldn't necessarily use it. But cells don't respond in the same way in terms of, because FBS is sometimes from a fetus, so there's way more growth factors and associated chemical signatures trying to push cells to grow, which is what people using it for cell culture are normally using it for. They want their cells to grow and continue to grow. When you're replacing that with adult human serum, you've got adults aren't growing so much, so there is less impetus for the cells to grow, so it's possible that cells will grow slower. Certain cells might be more susceptible to that. They might need that extra push from the growth factors. I also know that there's some cell types which human serum, there's something about it can actually drive epithelial mesenchymal transition, so it can change the cell type a little bit. So you might need to look at other alternatives for replacement rather than human serum per se. I don't think human serum will work for every single cell type. Hopefully that answers. I don't think we have a definitive answer for every cell type yet.
Thank you so much, Laura. That's a brilliant answer. My assumption was what Jeff put in the chat, which was just availability and expense. But I suspected there might be some more to it along the lines that you said. But that was really detailed. So thank you so much. Jeff's just put something else in the chat too, which is really helpful. Okay.
Sorry. It's fine.
No,
I will
please
reply in the chat. I was just
okay.
I've found that for my cells I've used the same concentration as I have for FBS of human serum. So not always, but sometimes.
Thank you very much, Laura. Really appreciate that. Okay, so time to move on because we're just about keeping time. So the next group of 5, we're going to start with Fiona, who investigated the physical and biological changes that occur within the body as a result of a heart attack and severe hemorrhage at the University of Nottingham. Welcome, Fiona.
Hello, thank you very much. I'm just going to share my screen. Okay. So hi everyone, my name's Fiona and I'm currently studying at the University of Nottingham in my 4th year of medicine. And I took part in a project this summer that used computational modelling or code to simulate cardiac arrest with the supervision of Dr. Morwenna Oldfield and Clara Dori-Vigna. And our project is basically looking at the heart. Sorry, one sec. Our project's basically looking at the heart, specifically the field of research that's trying to improve our understanding of cardiac arrest and the causes of cardiac arrest. And we found that many research papers use animal models, predominantly rodents or pigs, which involves, for example, inducing cardiac arrests on the animals by making them bleed out or by restricting the blood flow to their heart, which ultimately leads to their death. So in our project, we wanted to create an animal-free alternative to simulate cardiac arrest for researchers wanting to study it. So our summer projects use code that's been developed over the past decade by a team at Nottingham called the ICSM, the Interdisciplinary and Collaboration in Systems Medicine, that replicates the heart and lung systems in the human body. And we use this code and further develop this code to more accurately model what happens in a human when a human is beginning to have a cardiac arrest. And how we went about doing this is by firstly reading literature to guide our method. We knew that we wanted to look at the two causes of cardiac arrest, which is one, myocardial infarction, which is when blood flow can get cut off due to plaques building up in the vessels leading to the heart, and two, hemorrhagic shock, which is severe blood loss. And we brought in our knowledge from the literature reading to create a method that is relevant to research already out there so we can easily compare results. And what we ended up doing is, for the first cause of cardiac arrest, is we reduced the blood flow to the heart by 10 to 50% in simulated humans. So this is just increasing the resistance of the blood vessels in the code. And then we also added compensation mechanisms such as ventricular dilatation into the code, as the literature show that these are very important factors that occur in the human body when they experience myocardial infarction. And for the second cause of cardiac arrest, we removed the blood volume from a simulated human in varying degrees until cardiac arrest occurred. And the literature also showed important compensation mechanisms that occurs with blood loss, being the baroreflex, which we coded into the model as well. And the results were quite rewarding. When I retrieved the data from the simulations, we found that our results aligned quite well with what's found in literature. And what this means is that we're able to replace the animal models that uses a similar methodology to us. For example, one paper that looked at myocardial infarction used 39 large white pigs. Another paper that looked at blood loss used 13 dogs. So from these two papers that we've referenced, we could technically and theoretically save 52 lives. And lastly, an overall significance of having accurate computer models that it will be easier to study the process of cardiac arrest, which will be important when figuring out treatments for people who are likely or about to have cardiac arrests. And that's my summer project. Thank you very much to the facilitators at Animal Free Research UK for making this possible, as well as my supervisors, who have been very supportive, and they taught me a lot about the process of researching and creating novel techniques. And it's been a great opportunity to see what other students have been up to in the past 2 days. And yeah, thank you for listening.
Thank you so much, Fiona. The next presenter is Rosie Davis, who developed better human relevant models of bowel cancer to allow scientists to study how drugs penetrate denser cancer tissue without using animals at Sheffield Hallam University. Welcome, Rosie.
Thank you. Share the screen.
Rosie, you're just a little bit quiet. I don't know if you can fiddle with your mic again.
Is it a bit better now?
Not yet.
I tried to sit a bit closer.
Perfect. Yeah, that's worked now.
Okay. Hi, I'm Rosie. I'm a biology undergraduate at Sheffield Hallam. So bowel cancer is the third most common cancer in the world, and like most cancer research heavily depends on using mice as models. So the aim of this project was to develop a cell culture based model to replace mice and sort of generate the stiff fibrous tissue we see in where cancer grows. And the aim was to study drug permeability and see if drug uptake was affected by this dense barrier. So I was really happy because I got to spend time in a cell culture lab, which I really enjoyed. And I was working with a research team who had previously developed a collagen-based model where with fibroblasts, where you can grow cancer cells in, and they grow in clusters and form spheroids. And the middle picture there, labeled with the B, is an H&E stain of one of these gels that I made. And the larger dots are these clusters of cancer cells in these spheroids. And when you treat these gels with an enzyme called transglutaminase, it forms cross-links with extracellular matrix proteins. And this forms this fibrotic, dense tissue barrier, mimicking what we see in cancer patients. And then the idea was, because they're this 3D model, I could also manipulate them further with compression and try removing water, and to see if this had a further effect on the barrier. So we were using doxorubicin as the cancer drug, which fluoresces red, as in that center picture. So the picture labeled with D is just the cells stained with DAPI, just so we could see where the nuclei were. And then E is doxorubicin, and the F is the merged image. And so what I did was I tried various concentrations of the doxorubicin, as well as different lengths of time that the cells were exposed to it. And the idea being that we're trying to optimize this assay so that we could use it to test the permeability of the membrane to doxorubicin. And so the next step, after we found out that it actually worked and we could visualize doxorubicin, these images are after 24 hours with 50 micrograms per mil of the doxorubicin. The next step was to then try inhibiting the transglutaminase or treating with it, and mixing that with compressing the gels, all sort of either trying to build this barrier or inhibiting the enzyme would go from a different angle, just like removing it, and to see the effect of this on drug uptake. So my next step is to analyze all that data and see if it's affected the doxorubicin. And what's really interesting is that it was successful. There is an assay that you can do in the lab to see how much doxorubicin can be taken into cancer cells without the need for mice. And it'd be really interesting to see what the data tells me about the different effects of transglutaminase on it. And I really loved working in Cell Culture Lab with the research team. I just had a great time, and knowing it was for such a wonderful cause and the possibility of helping animals was just a really lovely thing. So thank you.
Thank you very much, Rosie, much appreciated. Next up is Imogen Carmichael, who tested if an animal-free media can be used to grow and differentiate a cellular model of dementia at Keele University. Welcome, Imogen.
Hi, I'm Imogen Carmichael. I was studying a new cell-based model for looking at dementia at Keele University. I just wanted to start by giving you a little bit of background into why this research was important. Traditionally, FBS or fetal bovine serum is used to grow eukaryotic cells, and it comes from the blood of dead calves, which is something that we wanted to look at replacing with an animal-free media. And tau proteins are found in brain cells, and phosphorylated tau is used as a primary biomarker for Alzheimer's disease, which was what we were looking to see in the cells that we grew. From there, I wanted to just give a simple walkthrough as to what we looked at. We started by growing SHSY5Y cells in 2 different types of animal-free media, brain fizz and knockout, which were 2 very interesting replacement medias. Great fun to use. Cell culture was quite interesting, trying to get them to stay alive. We did have a couple of issues with some of them, but they did eventually get to 70% confluence, which was where we were then able to start detaching them. We passaged them every 3 days in a ratio of 1:5. And eventually the cells were able to be seeded and differentiated using retinoic acid, which helped promote neuronal differentiation and maintained the neurophenotype. DMSO was then also added to monitor cell viability. And finally, oxalic acid was added in different concentrations to induce the hyperphosphorylated forms of tau in the cells, which was something that we really wanted to produce. We used a couple of assays to look at how well we were able to do this, starting with an MTT assay, which was to just study the cell viability. We then did a reactive oxygen species assay to study the cellular stress, and finally an in-cell enzyme-linked immunoassay or an ELISA to study the expression of the tau that we had formed. And if I just draw your attention to the table that's just popped up, that was how we were able to compare the two new types of media. When we were looking at the cell viability, it seemed that both were fairly comparable. It seemed that the knockout slightly beat the brain fizz in terms of how well the cells were growing, but it was still very close. When we then looked at the Ross assay, it showed that the brain fizz actually resulted in the cells being under a lot more stress than the knockout. But when we came to look at the formation of the p-tau proteins, brain fizz produced far more than that of the cells in the knockout media. So it is a very good way of replacing FBS as a general media. Both worked really well, but we think we prefer the brain fizz. And to sort of summarize, we were able to successfully grow SHSY5Y cells in 2 different types of animal-free media. The cells both managed to successfully produce p-tau tangles in both types of media as well, which was really nice to see. And we were able to compare the results that we got from both the animal-free medias to literature where they used FBS, and it goes to show that both do produce very similar results to the traditional media, which means that it is a nice way to easily replace animal-free, animal-produced media from the start. And I think that the next step would be to look into how we can then replicate this again to see if it really does continue to work and how we can then use it to look further into dementia research. And I've just got a few pictures of the cells that we were able to grow, which was quite nice. They're at 40 times magnification, which is a shame because we would have loved to get them a little bit clearer. But the lab didn't have a good enough magnifier at the time, so we were a little bit limited, but it was really lovely to see. It was an amazing opportunity to take part in. I really enjoyed it. It was something that I had never even known was available to me at this time in my degree. And I really enjoyed it. And I want to say thank you for letting me have this opportunity. It was amazing.
Thank you very much indeed, Imogen. Thanks ever so much. Okay, next, penultimate speaker is Yutong Chen, who explored state-of-the-art artificial intelligence-based methods to reduce the time taken to acquire magnetic resonance images, MRI images, at Imperial College London. Welcome, Yutong.
Hello, everyone. Today, I would like to introduce you to my summer project on MRI. So MRI is a high-resolution medical imaging technique on most of the soft tissues in the body, and this is an example of MRI of the brain. And the reason why I chose on working on MRI is because it has potential of replacing histology and biopsy of animals, which will require sacrificing animals. In MRI, it is non-invasive light imaging technique, which means that we can possibly use the same animals without killing them, or we can even directly apply on human studies. And however, despite this benefit of MRI, the limitation is that it takes a long time to acquire. So patients or animals may need to spend a lot of time in the scanners, which may create phenoclaustrophobia, and the prolonged time in the scanner also increases the cost. So I chose to work on compressed sensing. which is a technique to accelerate MR image acquisition in completely computationally. So hopefully I do not need extra hardwares, which may increase expense. So let me explain what compressed sensing means. So imagine that this full resolution MR image has 1,000 pixels. Compressed sensing only sample 500, for example, and therefore halving the scanning time. And then it will reconstruct the missing 500 pixels from the existing 500, and is performing extrapolation, but at the expense of reducing the scanning time. But the problem of this extrapolation is that it may produce blocky artifacts, and the image may become artificially smoothened, which may reduce the quality of diagnosis and may mislead doctors and researchers. And I was thinking if AI can help. So AI is I tend to understand AI is a computational method that mimic how our brain works, how our brain process information and learn and perform different tasks. An example of AI is generative adversarial network. So this network can be taught to generate photorealistic images like these shown here. So it is almost like a real human, but they really never exist. So I was quite impressed at the ability of AI to generate photorealistic images, and I was thinking if it can be applied to improving this recovery and reconstructing photorealistic MRI through undersampling. So let me explain how I apply this generative adversarial network. So it consists of a generator and a discriminator. So I tend to think of the generator I constructed as a painter or an artist that paint or try to reconstruct the original image and try to paint the picture. And I tend to think of the discriminator as a critique of the artwork generated by the generator. And it is through this critique that the generator continues to improve its reconstruction accuracy so that it can perform accurate extrapolation at the while reducing the MR scanning time. So example of the structure of the generator is shown here. It is not trivial to get this the structure of the generator that eventually works. It is akin to let's say wiring of the neurons in the brain. So I find obtaining this optimal structure to be a trial and error process. But I was really satisfied when finally obtaining a good generator that successfully and accurately reconstructs the undersample MR images as shown here in the last column. So as you see, the AI generated images are significantly clearer and is closer to the original image here compared with those generated by traditional compressed sensing method with higher resolution and fewer artifacts. And I was really happy that theoretically it can achieve 10 times the acceleration compared to the original MR scanning method, which means that if you need to be scanned for an hour in the scanner, the scanning time will reduce to, say, 6 minutes. And I would just like to emphasize that the technique may not directly replace animals, but instead it is offering a non-invasive alternative to animal research. So we do not need, by promoting MRI, perhaps we do not need to kill animals, do not need to sacrifice them to biopsy. We can directly perform light imaging of them and to reduce their suffering. And ultimately, I hope to reduce the suffering of both animals and humans through my research. And thank you for listening.
Thank you much. Thank you very much indeed, Yutong. Okay, last but by no means least, Dania Al-Khatib, who used artificial intelligence approaches to address an important unmet clinical need. Can we predict tumour recurrence for head and neck cancer at Queen Mary University of London? So welcome, Dania.
Hello, good evening, everyone. I'll be sharing with you my research project, which is entitled as Validation of a Cancer Stem Cell Prognostic Score in Head and Neck Cancer using Machine Learning Approaches. But before that, just a quick introduction about myself. My name is Dania. I'm a dentist, and I've done my master's degree in experimental oral pathology at Barts and London. I'll be sharing this project, which was done under the supervision of my supervisor, Dr. Jihad Youssef, at the ARC, the Animal Replacement Center of Excellence, which is actually funded by the Animal Free Research. So I hope you enjoy it, and I hope you find it interesting as much as I do. So cancer is considered to be the second leading cause of death worldwide, and head and neck tumors or head and neck cancer is considered to be the sixth most common cancer on a global level. Until this moment, there is still no clinically applied way that can predict the likely outcome of head and neck tumors, whether it's going to be a good prognosis, which means there will be no recurrence, no metastasis or spread within 5 years of treatment, or there will be poor prognosis, which means there will be recurrence or metastasis. And so in this specific project, we thought of predicting ways that could help us do that in a computational manner using AI and machine learning approaches. So we started by working on metrics that work based on transcriptomics that quantify EMT scores in 202 head and neck cancer samples that were taken from humans. And these metrics are the GS and the MLR. So we tried at first to understand why EMT is important as an indicator for the prognosis of cancer. And it was found that it is actually a strong indicator because it plays a role in metastasis, it plays a role in the cancer invasion, and it has an actual effect on the tumor resistance to drugs. So we started by finding the relationship between these two metrics, the GS score and the MLR, which was found to be a negative relationship. So as the score for the GS is increased, that means that the sample would be more enriched with epithelial tissue. And as the MLR score would be increased, that means that the sample would be enriched with mesenchymal tissue. So we started working on a way that would allow us to get a high prognostic accuracy using these metrics. So we did that using different classifiers, which are machine learning algorithms that categorize data into different samples. And we started using all of them for the GS, for the MLR, and for a combination of GS and MLR in hopes that it would lead to a higher prognostic accuracy. But unfortunately, it didn't. What we ended up with a 62% prognostic accuracy with the MLR score while using the K-neighbors classifier. 62 is very good, but it's not excellent. So we aim for excellence. That's why we aimed on expanding the MLR score using a correlation with more than 60,000 genes. And of them, we ranked them, and of them, we have chosen the top 40 best correlating genes with the MLR. We went on and did more computational methods that when we did a correlation between all of the 202 patients, which we already know before the prognosis of all of them, whether it was good or bad, we found out that after validating the true values of this correlation, after using obviously a 70% threshold of the correlation, we were able to get to a prognostic accuracy as high as 80%. So we started with 62% and we ended with a prognostic accuracy of 80%. And that by itself, I believe, is a success. And we were able to do that without jeopardizing animals, without using any animal models, which is another success. And that was actually the main aim of the project. Thank you so much for listening, and I would like to thank Animal Free Research Organization for providing us with such opportunities. That through this opportunity, I was able to learn more about a novel aspect of research and science, which is machine learning, that I've never encountered before. And in the same time, it gave me a new insight that I can actually progress with research and science without harming animals and without using them. So thank you again, and big thank you to my supervisor, Dr. Jihad Youssef, for his kind guidance and his obviously amazing scientific knowledge throughout this summer project. And I'm happy to take any questions you have.
Thank you very much indeed, Dania. What a way to finish. So that concludes our presentations. And as we did for the last group of 5, we'll take 10 minutes or so for questions to these 5 presenters. Mimo, did you collate them? Here they come.
Sorry, 1 second. It just seems like I cannot cut a paste anymore. There you go.
Possibly we could start the ball rolling while we're waiting for what a really important point that came up in the chat, and that is being discussed among scientists worldwide. It's a big issue, not least by a paper by Don Ingber at the Wyss Institute in America, which is world famous. Why are people still requiring validation in an animal model? You may have seen that I put in the chat, it's unscientific. You can't validate something new, a new approach, against a model that is demonstrably poor. Otherwise, no matter how good your new approach is, it's going to look poor because it's not going to agree with the poor model that you're using to validate it against. And this is frustrating more and more scientists, I think, and hopefully we're going to reach, this is going to boil over and people are going to demand some change. But I just wondered if, just to set the ball rolling, if anybody would like to chip in on this and let us know their opinions. Amanda, would you like to start, because you mentioned that in the chat briefly?
Sure. I used a metaphor, as I do, of the Hoover Dam, which I really do feel like there is this swell behind this barrier that we all experience, and how pervasive it is. And it's not just in terms of us getting published, doing animal-free research and actually being able to say, listen, this is a real thing. But it's also regulatory. It's also at the level of government. And I was just wondering how our summer students felt about this, and how we could maybe train their eyes. We're a little bit jaded by this. It's been many years, and we all know that once we break this dam, there's going to be a swell. It's going to be an absolute flood of all new treatments and new approaches that will come behind it. But currently, we are being asked to validate everything from human models to computer models through machine learning. Everything has to be put back into an animal in order to validate it, which makes absolutely no sense. I think I would use the word bonkers. So I just wondered how our summer students felt about that, and if they could actually see a very clear way forward that we haven't spotted yet, because fresh eyes are often sometimes the best. Not to put anybody on the spot, of course.
Thank you, Amanda. Before we go to the students, and I think this is a great discussion, and hopefully you'll enjoy weighing in too. Nick, would you like, because you brought this up as well, would you like to say something? And also Valerie, would you like to say something quickly before we go to the students too?
I can't really put it any better than Amanda just has, to be honest. I think that was a fantastic way of putting it. But it is a question that we are asked. If we are seeking funding to do biomedical research, it is a question that we're asked, and it is, the Hoover Dam analogy works. It is like banging your head against the wall to try to persuade people that what we're arguing for is a better way of doing it than the animal experiments. And how it becomes a very circular argument. It's we're going around in circles. So I can't put it any better than Amanda. I think that gets to the heart of the matter. I don't have any solutions. I don't know what to do other than keep making the argument, which it appears I'm not making convincingly enough at the moment.
Thank you, Nick. Valerie, would you like to say anything?
Yeah, if I don't seem to be able to start my video, but there we go. Maybe that's it now. Yeah, I think Amanda made the point very, very well. It is a problem. I think though we are gradually chipping away, and having, you know, the young people that have been doing the summer projects and getting this embedded at the very start of their scientific careers is one way that we can really start to challenge what has been accepted for too long, in my opinion. And it's good to see that organisations like the MRC, who recently had a call for non-animal methods, are gradually getting round to that way of thinking as well. So the Hoover Dam may well be about to be breached. Let's hope it's not too long.
Thank you, Valerie. If I can quickly mention something, and I think this maybe did come from Don Ingber, actually the author of the paper, who said that when they were developing these amazing organ-on-a-chip approaches, I think it was particularly regulators, the drug developers, were saying, well, that's all great, human on a chip, but we want a dog on a chip, and we want a rat on a chip, and a mouse on a chip. And his initial reaction was, why? Why do you want this when you've got human organs on a chip? Well, we just do because they're the species we use. So so we want the chips. But he said that actually what providing those animals on a chip enabled them to do was to look at how human relevant the human on a chip approaches were, and then to directly compare what they were finding on the human organs on a chip with the rat, the mouse, the dog, etc. on a chip, and to see right in front of them how different the results were. And in one fell swoop, that tells them you need to use the human on a chip because it's different to the other animals on a chip, and it's the most human relevant you're going to get. So I think, you know, this is how we're going to chip away at it. This is how we're going to change things. And again, and I use the phrase all the time, but I think it's important. We will reach a critical mass, if you like the Hoover Dam analogy, where the dam breaches and it goes. And we are heading towards that. We're heading towards that more quickly, I think. And this is becoming more mainstream, and there are more very strong scientific voices behind it. So that's I think that really is some cause for optimism and encouragement. But out to the students now. Shall we go through the last 5 speakers? So Fiona, would you like to? Well, actually, Fiona, there was a question specific relevant to this, a question specific about that in the chat. Would you like to comment on that?
Sorry, is that the question by Jihad?
Yes, yeah.
Okay, so thank you for the question, Jihad. We actually tried taking a step further to validate our computational model without the use of animal models, such as trying to find case studies of people who had hemorrhages and looking at the data to see how that compares with the data that came out from our simulation. So I think that's one way we could go about it, but in our project we didn't have enough time to find a comparable case study. Nevertheless, I think our computational model is moving towards being more accurate to like simulating humans than what animal models can do, because I did read some things from the literature model that, like, for example, small mammals, they have different compensation mechanisms when they have myocardial ischemia to humans. I think they reduced their energy expenditure or something like that. So whereas our computational model, we can like gear it towards an average human, like a 70 kilogram man. So yeah, I think there is work to validate the ICSM computational model. And I think with the team at Nottingham, it was quite nice to see how it's become more accurate with lots of little projects. But yeah, I think that's all I can weigh in at the moment.
Okay, thank you very much, Fiona. Would any of the last group of 5 students like to, because we need to move on a little bit, but would any of you like to comment on, I think it was one of Laura Jane's points, which was, do any of you have anything to tell us of the reaction from your peers about what you're doing and about your attitude to to animal-based research and to human-specific research. Is there anything you'd like to share with us along those lines, good or bad or indifferent?
Yeah, I'll say something. I think the response I had from whenever I told any friends or relatives about what I was doing over the summer, I don't think any of them had heard of animal-free research before. And I think they were just like in awe of it, really. And they just said it sounded wonderful what I was doing and the whole thought behind it and the reason why. It was all just, yeah, really positive.
That's great to hear. Thank you. Thank you, Rosie. Dania has her hand up. Dania, would you like to say something?
Yes, I'd like to say that especially with the prognostic score that I mentioned in my project, most of the ones that happen now without using computational methods are done on dogs, whether we're talking about cancer, whether we're talking about renal problems or even brain injuries. And dogs, to many people, among myself, I considered best friends. So they were really happy, like my friends and all, they were really happy that I was able to save dogs, a lot of dogs, from having to suffer through such experiments and finding an alternative way.
Thank you very much. And Fiona, you have your hand up too.
So when I told my friends or other peers in my course about it, because they know that I'm choosing a vegan lifestyle over the past year, and they thought that a scheme like this is like really resonated with my values, and because I told them recently about, like, in the news there was it Cruelty Free International, they uncovered the way that they would treat animals in the lab, and my friends were quite shocked about that. So when I told them about this opportunity that I get to work with Animal Free Research UK, they just thought that it was a really good opportunity for me.
That's great to hear. Thank you very much, Fiona. Izzy.
Yeah, so just relating to the debate that was going on in the chat and that you mentioned, I think that it really depends on the type of person that you want to connect with about the issue of animal research. So for someone with no scientific background, I think it's actually really useful just to strip back to the basics and ask them, how do you think that researchers research cancer? They can't. I've asked it myself to people with no scientific background and they couldn't answer. They were just like, oh, I don't know. And you think, well, you have to give an animal cancer, and they're shocked that, you know, and they're like, oh, of course, that's like how you would research it. And I think, yeah, you have to give an animal cancer, and then ultimately they will be sacrificed. And they find that really emotive. And by saying that, I didn't have to engage in any scientific knowledge. I didn't have to communicate any, you know, technical language or anything. I just said, you know, these animals are being sacrificed, and you know, that's how it's done. And people say, you know, oh, I think for cosmetic purposes animals shouldn't be used. And I think, right, great, good start. And then they're saying, but, you know, if there's no other alternative, then they should be used. I think, but there is alternative. And they go, really? So I think there's just not enough awareness of the fact that there are alternatives to this. Yeah, I think it is completely the publicity and the way that the way that you can reach different members of the public it's got to be tailored slightly differently, because there's no point obviously just pumping out scientific knowledge to people who won't be able to understand the science. So just connecting with people on using emotion, really. I mean, it kind of sounds a bit brutal, but it is using emotion and saying, like, people don't know that scientists do use dogs, and it's just they do, but nobody knows. So as soon as you say, oh, you know what, actually 60 dogs were used in that experiment, they'd be absolutely mortified, because you can relate to the fact that people have pets of their own. Yeah, I think it is a really interesting issue, and I think that the way that to get messages out is really hard.
Thank you, Izzy. Thanks for all of your input. I think just to very quickly give you my thoughts before we move on. This is, you know, I left laboratory research 17 years ago, I think, and I've spent that 17 years doing this for various groups, trying to really educate people about the science, why we do this, why we don't do that, what we need to do, what we need to stop doing. And it's such a complex field, but I think, you know, there are times where the information has been out there, where we've got a huge media article and you thought, great, this is going to reach so many people. But as you said, Izzy, I remain surprised by how much, and this is not having a go at anyone. I think it's largely because it's difficult to get the word out there. The press tend not to be particularly interested, or the public read it and then forget about it. But so many people just don't know. They don't know what goes on and why. They don't know the degree of failure of a particular approach. They don't know about what science is generating instead, and that there's a better way forward. There's just people just don't know. So our job, try as hard as we might, is to try and educate everybody about this. But ultimately, you have to use certain avenues to do that. And if they're not as willing as you would like them to be, or as they should be, to help get the message out there, then you're going to struggle. And I think that's where we are. But I think science is becoming more exciting. And as we really start to see the shift that we've spoken about and the success that comes from that, again, hopefully, the Hoover Dam analogy, it will just fall and out it'll come, and that will be the way things are done, and we will wonder why animal research was ever the gold standard, if you like. So I think yeah, there's lots in the chat, we can read it later, but I think we really need to move on to try and keep time. So Carla, I think it's over to you.
Thank you, and brilliant timing. My dogs all just started barking, so hopefully you can't hear them. Thank you so much to all of our students. You've been absolutely brilliant. Well done. You've really presented your work well, and it's so inspiring to see the next generation of scientists who are actually going to make this change. So we are going to get this done. It feels frustrating at the moment. We are going to change this in our lifetime. So it's going to be down to you guys. So no pressure, but yeah, please end the use of animals in medical research once and for all. We're all counting on you. So I'm going to ask Amanda to say just a few words about in celebration of what you've all achieved. To speed things up slightly, I'm going to also ask you, while she's talking, to open up your parcels that hopefully all of you, maybe not Dania, but everybody else has hopefully received, which has got your certificates in. And then while Amanda's speaking, maybe you can do that, and then we'll ask you to all hold them up and we'll get a photo of you. So over to you, Amanda.
And I'll speak fast, as is my way. So tell me to slow down if you need me to. Can I just say, there's nothing that I can say now that can top what we've heard here already today. I've been so impressed with the quality of all of you, each and every one of you, and the quality of discourse and debate that we have had in the questions and answer sessions. And I say this not lightly. I have been at many a boring conference, and this is not one of them. So thank you for a couple of hours, very, very well spent, that has fed my intellectual development. I've been sitting here thinking back to what I was like and what I was doing when I was at your stage. And okay, all right, that was the last century, but even still, let me tell you what the differences are between now and then. So in my day as a student, sure, there was social protest against lots of things. I was in Ireland in the 1990s, and of course there were a few kinks to iron out, so there's plenty to protest about, but of course animal welfare protest was in the mix there. It was also cool to be found face down in a bar on a Friday night. That's pretty cool then. It wasn't necessarily cool to be a scientist, and being not intrinsically cool as a person, I had found my niche. I'd also never ever gotten past the why phase, so it suited me just about fine. But something that's happened since then is that it has become cool to be a scientist, to be a geek, to be a nerd. These are badges of honor that we hold dear and display proudly. It has become cool to ask questions. It has become cool to change the world, not just by talking about things and shouting about them on the street sometimes, but by actually doing something about it. And this is what you have encapsulated today. So one of the reasons why we have our studentship scheme is to engage young people with a problem that exists not just for our animal partners in this world, but humanity at large. And as many of you mentioned, there's a huge lack of awareness, not only about the number of animals that are used in clinical and biomedical research, but also around the alternatives that currently exist or could exist with the judicious piece of research, like you have done. So this is where you come in, and you now know that we have your back. You have helped us to realize our vision, epitomizing the backbone of our values, and we are behind you all the way. And behind us are our wonderful supporters out there who share our aims, and with them and with you, we know that our aim of removing the need for animal use and research can and will be met. So you will now walk on in your life's journey, changed forever, with knowledge as your tools and experience as your shield. So go out there and seed the world with your ideas. You are our ambassadors, and you are carriers of this hugely important message. You are our future. So go forth, use your powers for the forces of good, for animals and for humans. And I want to thank you for all of your hard work and wish you all the best of everything in your futures.
Thank you, Amanda. And let's have a round of applause for all of our students. Well done, guys. Can I ask, I'd really like to get a photo, so a group photo. We'll try and see if we can do this in a Zoom context. So can everyone who isn't a summer student turn off your videos? I'll do mine as well. And then can all of the summer students keep yours on and hold up your certificates, and Jess can hopefully work her magic and get a group photo. Fingers crossed this works.
Not showing everyone on one screen.
That is a shame. Okay, I think what summer students, you all need to speak, and then that will shift you to the front.
Hello.
Oh.
Hello. Hopefully this works.
You can also drag people around, I think, if that helps.
You can.
Oh, you can. Thank you.
Izzy, you're a genius.
Okay. Everybody ready? Some smiles.
Yay, congratulations.
Well done. Brilliant. And can we try and do a group photo for everyone who's here? So can everyone put their videos on? Only if you want to be in the photo, of course, but if everybody could put their videos back on, and we'll do a group photo of everybody.
We need to
If
move.
we were all together in the physical room, hopefully next year. Jess, you can maybe count us down for when you want us to.
Okay. I'm gonna have to do a couple because again, it's not on one screen. Okay. Eyebrow smile. And keep smiling. Great, thank you.
Wonderful. Thank you so much, everybody. So here we are at the end of another great event, just about on time. So thank you all for joining us today. Thank you to the students. We really hope that you continue this journey. You are the future, and you're going to be the ones who finally make this change. Thank you so much to their supervisors who've expertly supported our students in learning pioneering animal-free research techniques. It's really quite unique. I don't think many undergraduates get to do this. So thank you for supporting them. And thank you to all of our amazing supporters here today. I hope you feel as proud of our students as we are. And I hope that you'll continue to support the next generation of animal-free scientists. Thank you. I'll hand over to Jared for a few extra words. Mimo, yes?
Yeah, no, there was another thing in the package, some I received, I believe. Is that correct, Darcy?
Yeah. Oh, Fiona, you're holding it up.
Something else. So if you want to show it, if you got it, Yeah, great. Very pleased. So all the students got a special prize. They got a lab coat with our animal-free research. So wherever they go, they can continue being our ambassadors. I'm proud to be. Great, thank you.
I think they should put them on, and we have a photograph of your own as well.
Sure.
We're hoping that you'll wear them back at university in your labs to help raise the profile of Animal-Free Research UK.
Send us a selfie with that.
Dania, yours is on your way. It's on its way to you. Yeah, please send us a selfie. So in the interest of time, Jared and then Laura Jane, love to invite you to just say a few words in closing.
Okay, thank you very much. The first thing I want to say is I'm very jealous of the people with the lab coats, because I don't have one and I would really like one. So so you never know. Yeah, so look, just once again, it's all been said, but just to say it again, thank you so much to our incredible and ever-growing community of animal-free scientists. I think represented here today by some of the highest achievers, a real example that animal-free research is not only possible, but is the only way forward, the only way we can really make a difference. As I said, for hundreds of millions of animals every year, and for the 7 billion people on this planet, and what they need science to do for them. So thank you so much. It's been a real pleasure to spend a little bit of time with you over the last couple of days, and all the very best for the future. But if the students could please stay behind for a little debrief, we'd really appreciate that. Not for long, so please do that. Thank you. And Laura Jane, please say a final few words. We love that. Thank you.
Oh, thank you, Jared. There's nothing I can say that has not already been said. We're so proud of every single one of you. Hearing your presentations was just incredible, and the chat that's been happening, the questions that have been asked, and the way that you've all contributed to all of those conversations is just, it's really, really encouraging. It gives us such faith that this really, really can be done. So just massive well done to every one of you. You should be so, so proud of yourselves. And I guess just again to say on behalf of the trustees of Animal Free Research UK, we're volunteers. We don't get to see this kind of thing very often. So it's just absolutely wonderful to, even though it's not in person, to see your faces and hear directly from you about the amazing achievements that you're making. So yeah, just congratulations again. Thank you for everything. And thank you to everybody for joining us today. Have a lovely evening. Thank you so much.
Thank you.
Thank you so much, everyone. Thanks so much for joining us. We'll see you again next time.
Bye.
Thank you, everyone. Goodbye.
Bye.
Thanks, everyone. Bye.
Bye.